waiting in the queue. And I was
Mhm.
touched actually by a message I got on my social media channel from an actual organ transplantation patient who just like
Mhm.
begged me to work harder and faster because he didn't have that long, I mean,
Mhm.
left according to statistics. It is known that after receiving organs, transplanted organs, even though your life is extended, but it's still
Mhm.
there's still a limit of how long that organ can support you.
Hey everyone, welcome to It's a Material World. We're the show that uncovers why material science will change the world. Consider subscribing and hitting the like button down below. It would really help us out. And before we get started, just wanted to mention that we have released material science merchandise for those of you who want to support us or just express your passion for MSE. You can check out more of the designs at itsmaterialworldpodcast.com/shop or by clicking the link in the description. All right, let's get right into it. Hello, everyone. We are super excited to welcome today's guest, Dr. Itedale Namro Redwan, onto the show. She is the Chief Scientific Officer and Director of Bioink and Tissue Engineering R&D at CELLINK, which is a bioink company that aims to revolutionize 3D bioprinting for pharmaceuticals and drug development. There is massive potential in this field, so we definitely have a lot to discuss today. Thank you so much for joining us.
Well, thank you for inviting me, and I'm super excited to be part of your show today and hopefully inspire some people.
Of course.
Awesome. Yeah, thanks so much for joining us. So I guess let's start at ground zero. What exactly is 3D bioprinting and how does it differ from traditional 3D printing?
Yeah, so bioprinting is an additive manufacturing process similar to 3D printing.
Mhm.
But instead of printing with plastics, we're actually printing with cells and biomaterials. And we are trying to create organ-like tissue structures. So we let the cells live in the material and we try to mimic the native tissue as much as possible.
Wow. So what kind of materials are used? Because you mentioned plastics are used in traditional 3D printing techniques, but I still believe there's like biopolymers potentially, right, within 3D bioprinting?
Absolutely. So, I mean, what we try to do, of course, to be able to print hydrogels and biomaterials, you need the polymer part of it. And we, of course, get a lot of inspiration from nature. So many of the traditional hydrogels comprise of collagen, gelatin, alginate even. But also in the end of the day, what we try to do is we mimic the native environment by inspiration of the native tissue. So maybe by having collagen, we try to mix in ECM components within that hydrogel to mimic the native environment.
Interesting. And so when you bioprint, I think we're all familiar with what 3D printing looks like. You extrude plastic and you build up from the ground up. What does 3D bioprinting look like? Is it a similar method or does it completely revolutionize like how we would think of 3D printing?
No, I mean, this is why it's a additive manufacturing approach. It is very similar. The big difference here is that we are building a tissue construct layer by layer. I mean, if you look at the concept, it actually starts exactly the same way. When a scientist wants to bioprint, they actually need the G code or the STL file, which is the, you know, computer map that tells the bioprinter this is what you need to print, where, how much, how fast, and what. In what position. So it follows the same theory. However, with bioprinting, you're actually mixing cells with a biomaterial that is printable, and you're able to print, literally print, layer by layer, living cells.
Awesome. I'm super curious about how those two are integrated, but we'll get into that a little bit later. First, I just want to start with the applications that 3D bioprinting offers. So I know it ranges from like functional hearts, albeit not fully sized just yet, to skin for burn victims. And so I was just wondering, in your experience, what has been the most interesting or impactful application you've seen so far?
Wow, that's a tough question. And as you might imagine in this field, and of course there are many, many favorite applications, but I do believe that the ones that directly help to contribute to the future of health are my favorite ones. For example, scientists within the field know that vascularizing large tissues is still very tricky to achieve. And using bioprinting, we can actually incorporate channels to provide the core of tissue with oxygen and nutrients, as well as, remove waste products. These more advanced 3D bioprinted models with, let's say, complex vascularized tissues will actually be useful for minimizing animal models in clinical trials. Two, they can actually help scientists to understand human diseases. And in addition to that, they can be used for drug screening purposes. So using bioprinted models, you can actually contribute to very different application areas. And I think anything that helps advancing the future of health or treating human diseases is what's really important to me. If I can mention one more, something that I think is also very important in addition to minimizing animals in clinical trials, I think personalized medicine. We know that we treat people in the same way. If you go to a doctor, you get the diagnosis with a specific disease, then you get the same treatment as a patient with the same disease, maybe in other areas of the world. We know our genetic background, gives us let's say, a differentiation in how we respond to drugs. So personalized medicine will actually allow us to derive the drug, identify the drug to treat that specific patient, and even identify to which dose. So that's also a favorite one.
Awesome. So the applications you just mentioned kind of had two purposes. One of them was almost re-engineering something that's already done commonly, which is like testing for cosmetic and other things, like the animal test subjects could be removed, that we do very commonly now. And then another one is the personalized medicine, which I think isn't used very much currently. Which one of these applications do you think it's ideal for? Is it replacing what's already there or completely revolutionizing a new part of medicine?
This is exactly it. I mean, with more advanced models, you can actually advance how we are solving future healthcare problems. Another problem that I see, I'm just giving more examples rather than answer to your question, but another one is definitely the medical device industry. What if you could use the patient's own cells, print them in the appropriate material, and implant them, as for example, to a diabetic patient to treat diabetes? So you have insulin producing cells. So, I mean, I see that with more advanced technologies and, of course, the amazing scientific community getting their hands onto these scientific technologies, we can advance the way we are diagnosing and also treating diseases, but also solving future healthcare problems and today's problems as well.
That's amazing. I'm working in the medical device industry right now. So the last part that you mentioned about just like insulin producing cells was super fascinating. And it seems like there's just a lot of potential for advancement in this field, even though there are also some challenges that need to be addressed, but that's similar to any up and coming field, you know?
Yeah, no, I'm going to keep my fingers crossed for you. Let's see where you end up in the future, Puneet.
Awesome. Well, I think we really wanted you to touch on that part of changing like how we think about medicine. And in a previous call, you mentioned some eye-opening statistics that kind of put into perspective exactly how much of a need there is for personalized medicine, but also just 3D printing that could solve multiple issues across the entire medical field. We would love for you to like ask us some, and we can guess what the actual statistic is, if you have some on the top of your head.
Absolutely. So this question goes to you then. How many years on average do you think it takes to bring a drug from lab bench to market?
Mhm. Well, I know that for COVID, the vaccine came out incredibly quick, and I think it was like 1.5 years. So I would guess like 7 years.
Yeah. Usually that's like an exception, I feel like. So I
Yeah.
think from what I've heard, the average timeline is 7 years, but I would, since David already guessed that, I'll estimate a little bit more than that and say 10 years.
So you're closer, Puneet.
Let's go.
No, it does definitely on average take 12 years is the
Oh,
current
wow.
number.
Wow.
But I do
Mhm.
have to admit, and I'm very, very proud of everyone who was behind the COVID vaccine, but
Mhm.
definitely it has to do with the amount of effort everyone put
Yeah.
in that same research topic. Of course, it's
Mhm.
going to be faster at that point. Can I ask another question?
Yeah,
Sure. Yeah.
go for it.
Can you guess how many lives are lost every day waiting for an organ in the transplantation queue?
Gosh. Mm.
Hmm. My guess is 20.
20. Following Pernice's method, I'll guess 25 this time.
Oh no, David.
Oh no. Oh no.
So it is actually 21. But I mean, the way it's expressed is that, one life is lost almost every hour.
Oh wow.
Wow.
Waiting in the queue. And I was
Mhm.
touched actually by a message I got on my social media channel from an actual organ transplantation patient who just like
Mhm.
begged me to work harder and faster because he didn't have that long, I mean,
Mhm.
left according to statistics. It is known that after receiving organs, transplanted organs, even though your life is extended, but it's still
Mhm.
there's still a limit of how long that organ can support you.
Yeah.
So yeah, one every hour. That's quite a lot.
Yeah, that's crazy. I remember doing like a project on 3D bioprinting and this idea of providing organs for those who are on the waiting list, because there's just so many people who are on a waiting list and it's so hard to you know be next on. So it's definitely a very powerful story that you just mentioned, and I hope we can continue to make advancements in this space.
Mhm.
I hope so too. Definitely.
Awesome. Well, if there's no more questions for us, I think I took the lead overall.
Yeah.
We've been doing this a few times in various episodes, so David has to play catch up pretty soon. But just from the Bioinks perspective, what properties are desired in order to be an effective material for 3D bioprinting?
Yeah, definitely the list is long, and I know that people who are in the field might think a lot differently. But if I'm gonna focus on keeping it quite general and
Sure.
not go into depth, then I would say it's important for a bioink to have beneficial printing properties. So you need to be able to print it. And you also need the bioink to have the ability to protect the cells during the printing process. In addition to that, you need to provide the cells with the appropriate environment to behave as they would in the native environment.
Mhm.
Another thing that's also very important is for the bioink to be able to retain its shape post the printing process. And of course, this can vary depending on application, from application to application.
And so when you print living cells, I've done a little bit of research and it looks like the cells would replicate and actually grow. How is that aspect of Bioinks thought of when printing organs or basically anything with Bioink?
So I mean, the design of a bioink is, I would say, it's a work of a very interdisciplinary team, and I'm very, very fortunate to have amazing individuals on the team with an amazing, knowledge within the field. So you actually, whenever you have a Bioink development project, you actually have a list of many questions that you need to answer. And one of those is, what is the environment we're going to mimic? Which tissue type is it? What is the function of the cells in that environment? And the list goes on. So the process is quite complex. If you want me to go into it a little bit more,
sure,
I'm more than happy to. So, for example, you have to start by understanding the microenvironment that you intend to mimic. Next, you need to understand the cross-linking method you intend to use, because it's very important to mimic the stiffness of the target tissue. We know that the mechanical properties of the tissue you're actually mimicking serves a function. For example, the stiffness of bone compared to the stiffness of brain, that's a huge area. So you need to mimic that. Keeping the shape after the printing is also very, very important. And this is something that you can change with thickeners and by changing the composition of the physical parameters during the printing process. Another thing is you need to define for how long you want the bioink to remain stable in culture. How long are you running a drug screening process where you only need the model for a week? That requires a different cross-linking method compared to having a culture that you want to grow for, let's say, 3 months plus.
Oh, wow.
I think the last but not least, something you need to define early on is whether the ink you're developing is intended to be applied in clinical applications in future, or be a translational ink, as we call it, because then you start in your design, consider GMP compliance and how you can get there in the future. And as you can see, with all of these properties, developing an ink is a trial and error process, but we learn from every error we do make. And this is really what we do here. And I have to add that you need amazing end users to evaluate the ink to also give you feedback.
Wow. Yeah, I was just about to ask you, it seems like if there is an extensive list of requirements for properties, there might not be enough materials right now to achieve all of those applications. So I was just wondering, what are common materials used in this space right now that at least can meet some of those requirements?
I would say that every tissue type requires a very, very careful thought process behind it when you're defining the ink, or let's say designing your bioink, so to say. Many of the very, very common materials, of course, I'm gonna brag about our patented cellulose as a base, because it's a sustainable material coming from wood and the nature. But some of the very, very common materials being used today are gelma, or let's say methacrylated gelatin, and the colma, which is methacrylated collagen. But alginate is also very, very commonly used. The field has exploded recently, and that's least to say. And there are some synthetic, semi-synthetic, but also natural polymers being used.
I guess going more into detail about an application you mentioned earlier was bioprinting of cancer tissue models would add that personalized medicine aspect where we could test individual drug trials on the cells in your body without actually harming the actual patient if the test wouldn't go well. How far away are we from this level of personalized medicine, and how can we help the development of biomaterials to get to that point?
So to be very honest, I do have a lot of collaborators worldwide working on this, and there is a lot of research ongoing in many, many labs. I do believe that we have advanced very much in this application area, to the point where it soon will be applied for diagnostics, but also for identifying appropriate patient-specific drugs. So biomaterial scientists are using a very different approach here. And the most common is using biomaterial designed, intended to mimic the diseased environment. For example, you can use ECM components to replicate an ECM environment of a cancer tumor.
Real quick, what does ECM stand for?
Extracellular matrix, and I'm very sorry for not the
no problem.
For us the ECM is something we say every day, but definitely I mean the extracellular matrix is definitely a very, very important part and plays a very, very important role in the cell-to-cell interaction, cell-to-cell communication, and even disease progression. So biomaterial sciences, I mean, we do get inspiration from nature, and the extracellular matrix is part of the diseased organ. Yeah, I think that's what I can say about that.
Yeah, so I guess finishing up on the question I asked was, how far away are we, and what big steps need to be taken to have this practice be used in basically all cancer patients or all diagnostic?
So I think at R&D phase, we have shown that it's feasible. And I've also talked to, research institutions, which is the next step from the academic science lab, who are trying to implement it translationally in clinic. I would say it's starting to come there. We will see it in the coming years, maybe. What would make it available for everyone is the cost. Of course, and I'm hoping we are a company that really wants to democratize the industry. So we are famous for working with collaborators and customers to providing our technology and make it available everywhere. But if you can look at this treatment or maybe this personalized medicine area, what it would require for finding a treatment for a very specific patient, it would require you take that patient, take a biopsy, culture the cells in vitro, expand them, and then you actually bioprint them, perform the drug screening in certain replicates, and then you actually, once you've identified the appropriate drug, you need to identify the dose. I don't think, I mean, it is a costful process still, so I think privileged countries will implement it earlier than less privileged countries, to be very honest. But it's close.
Yeah, and so I guess we talked about this before, but you introduced the idea that cancer and sometimes diseases are all cast under a very broad range of a name. And so each individual person experiences cancer in a different way. So can you kind of explain a little more and maybe expand on that idea of why personalized medicine would be so revolutionary? Sorry.
Yeah, I think instead of experimenting with the patient, I can give you, I mean, cancer is one of the, I would say, application areas that has advanced the fastest within the personalized medicine. I mean, you get a cancer patient, not only do they get diagnosed with cancer, but then there is, of course, we have a lot of experience and knowledge and know-how within cancer, and the doctors, they are doing, a lot. But still, there's a lot of work being done where you have to experiment on which drug to use to treat the patient. You might have to remove the tumor, then treat them with laser, then figure out which treatment to use after that. What if you could already at the stage of taking a biopsy to identify whether it's cancer or not, immediately start growing those cells, and then identify the right drug from the first go? You wouldn't put the patient through the trauma of not knowing whether they would respond to the treatment or not. And of course, that would also help us put those patients on the right medications faster. So, I mean, it's all about saving lives. You're saving lives by putting them on the right treatment, on the right dose from start.
Wow. Okay, this just came to mind, but it seems like data science or AI or ML could potentially be incorporated into like that diagnostic assessment. Is that something that is being worked on right now in this industry?
Absolutely. I mean,
Wow.
the entire workflow is being automated and made very, very, very intelligent. Of course, this is the way we need to work. We need to be faster. And this is part of bioconvergence. Bioconvergence is the combination of advanced cell biology with artificial intelligence engineering to advance the future of health and future of medicine.
Awesome. So we talked a little bit about this. You mentioned even there's different cancer types that could lead to more personalized treatments. And then also there's just a variety of structures in the biological space, from hearts to skin tissue. So I was just wondering, you talked about the trial and error process, but can you go into maybe a little bit more detail of like the material selection process, how those requirements are defined for each type of biological structure?
So the biological requirements are definitely defined or identified through inspiration from nature and the native tissue. So let's say you want to create a bioink for a vascularized, like let's say endothelial cells, which are the cells which makes up the our vascular network. Naturally, you're going to look at that specific environment and see what are the molecules expressed within that area, which factors, which biological factors, and then you get inspiration from that. And then, I mean, you might start by having a few prototypes a few Bioink prototypes that you evaluate functionally. So you make a few Bioink prototypes, then you go to the lab, you print the tissue model you want to mimic, and then you test which prototype provided the most functional tissue. So it is a lot of trial error, but ultimately you're trying to mimic the native environment from a functionality perspective, super important, and also from a biological perspective.
And so from that perspective of so much trial and error, I guess there could be multiple areas of improvement within that. It could be the processing time, so faster printing, or maybe using ML AI to have better selection. Where do you see the most growth potential to be able to get better at this material selection or other like selection issues?
Definitely incorporating imaging and AI for analysis. AI for imaging, first of all, but also the analysis part. So, I mean, don't forget that it's a long way after you've printed and matured your tissue. It's a long way until you can actually get the answers to, does this express the right markers? Does it express the required proteins? If we could automate that and also have a component where you can actually get the data analysis, everything done in the same workflow. That's going to be an advancement. And I think, we're not sitting and waiting for someone else to do it. Definitely, this is something we are working on without saying too much.
Cool. Well, I remember reading an article about the first fully functional 3D bioprinted heart. However, it was only the size of a rabbit's foot at the time. The concepts of smaller constructs today, building up to a full-size construct in the future, seems to be how the field is trending on how to build upon itself. What are the limitations that we face that don't allow us to just print fully functioning human organs at this point?
So I will mention a few limitations that I think are holding us back from the dream of getting organs transplanted into humans and, for this dream to become a reality. I think the appropriate and FDA compliant materials is one of the problems. And we know that that process is very, very long. But, that is one of the hurdles we need to overcome. Also, the production of larger organs with appropriate vasculature that can bring oxygen and nutrients to the cells and even, bring waste products out of the tissue model. That is also a challenge. But of course, there's a lot of work ongoing within that field, producing multicellular models with the same complex architecture as the human organ. Don't remember, you not only need to place these cells, but they also need to function the right way. So, printing, you get the inspiration, you place them next to each other, but they need to function the same way. So it is a complex process we want to mimic. But I think the right steps are being taken by the industry and by the scientists out there to get there.
So you mentioned this a few times before, and it's something that I read in the past, but why exactly is like vascularization such a prominent challenge in the space? Like, is it very difficult to mimic and create like a system that mimics this in the body?
If you look at our organs, they are really made up of a very, very, I would say, impressive network of vasculature. But they're also made up of many, many million of cells. And every cell needs to get nutrients and also remove waste products. So it's very, very advanced. And that is what we need to mimic. And of course, the time it takes to print that network and make sure it matures and every single cell gets nutrients and oxygen, that is the complexity. So I think it's underestimated how complex the human physiology is, but it is a very, very complex system we are trying to mimic.
Yeah. And so I know it took like hundreds of years to fully understand what was in the human body. And I'm sure that we haven't discovered everything yet. So when you're trying to mimic it, do you think currently there's more learning to be done about how exactly the human body works? Or is it more R&D needs to be done to understand how we can mimic it and have the same relationship between cells that we just print versus things that are in our body?
I think we understand the human body quite well. I would say not fully, and then we will continue to learn from nature and how we are made up as humans. So I wouldn't say we know it all yet. That's a continuous process of expanding our knowledge. And yes, the answer is correct. We need to learn how to mimic what we already know and how to recreate it without losing the functionality. It makes no sense printing something that is not functional for the sake of it. It needs to serve as functional tissue model, either for drug screening purposes, it needs to replicate the diseased model, or for regenerative purposes to replace let's say, diseased organ or tissue type.
So a follow-up question then with regards to those challenges, what are your biomaterial scientists doing currently? Like, are they working on materials development? Are they just trying materials that are, or trying to improve existing materials in the space? What's going on there?
Without, of course, going into too much confidential details, we are constantly improving existing materials and constantly looking into what are the next materials that's got to be the future. This is, the work never ends. We learn every day from our trials and errors, and those errors needs to be implemented. So I do have an amazing R&D team working on biomaterial development full time. And all they do is further develop, improve, and develop new ones, characterize, evaluate, and going back. It's like a never-ending circle for them.
That's awesome. Yeah, I can't wait to see what you all come up with next. And that kind of leads us into this next question. What are the next steps for CELLINK and the bioprinting industry as a whole? Like, where is there that room for further advancement?
Yeah. So, I mean, we as a company, we belong to the BioCo Group. The BioCo Group is now a bigger family, and we are very fortunate to have 11 sister companies. All of the companies within the BioCo Group offer technologies, products, and services in various application areas related to advancing the future of health, sticking to our mission and vision. And for us at CELLINK, we will continue to listen to our customers, listen to their challenges, to provide them with the solutions they need. And since we do have 11 other companies in the family, we can have access to very, very advanced technologies, including imaging, single cell sorting, genomics, all the omics field, even in industrial applications. So we are very, very fortunate to having access to that. My team are focused on always providing the latest technologies, reagents, and materials to advance the science within the bioprinting industry. And I have to say, we are very proud, but also very, very humble about the fact that CELLINK is the industry leader within the field of bioprinting. And it's all thanks to our amazing customers.
Awesome. So one application that I'm curious about is that when you're building upon mimicking a human structure, has it been thoroughly examined, or is it in the horizon about taking your cells and then modifying them slightly, such as when we were talking about insulin before? Could we take part of their liver and then modify it so it starts producing insulin if they're type 1 diabetic?
Now we're into gene therapy and CRISPR-Cas9, and I mean, of course, the very, very amazing system that received the Nobel Prize.
Yeah.
It's not still very, very implemented in the personalized medicine field, I would say, and it's still, what would be required would be that you would take the cells from the patient. Let's say you would take some cells, you would reprogram them to induced pluripotent stem cells, which are the stem cells that you can get from adult cells from a patient. Then you can actually gene edit those cells and then differentiate them to the original tissue that you wanted to create, for example, beta cells that can produce insulin. And then you can actually print a medical device with these cells with the right biomaterials and use them as a biomedical device for treating that patient. But I mean, there are many things that still needs to be, I would say, corrected, and we need to identify the right compliances and all of that. Number one, CRISPR-Cas9 for editing cells. What happens in the human? Number two, do we have the right materials? And so on. Interesting.
And so you mentioned the BioCo Group. I believe that stands for bioconvergence, right?
Correct.
Okay. And so I was just wondering, so I can see that there is that common vision of changing the world of healthcare and saving patient lives. I was just wondering, just from the general standpoint, how do you plan on like working and collaborating with each of the other companies in that group, while also just being able to focus on your own growth as a company?
I mean, there are many, many reasons why we are all in the same company. There is a fine line and red line being here, we all work with the same sector of health. And
Got it.
there are so many roads crossing. Like I can be working on a challenge in my lab, and by having access to many other companies, you could just, through the scientists in other companies, put in a question in a chat saying, hey, has anyone worked with this cell type, or has anyone worked with this application? And you get direct experience from other people in other companies. Next, the technologies we have in the BioCo family, it makes so much sense because we've listened to the customer to build the BioCo Group. We've actually looked at what is it that the collaborators, a customer wants to do there. Well, they have a research question. They want to find the answer to it on R&D level, and then they want to step it up. And then they go to a slightly translational level, and then they are ready to apply it clinically. And that's where we have the industrial sciences. So we can actually take that journey with our customer, just because we're in the BioCo Group, because we provide the instruments for R&D, for mid-stage science, and also industrial applications. By the way, that's a business area within the BioCo Group. That's why, there is a very, very well thought through red thread in every acquisition that has led to the BioCo Group.
Yeah, I guess another question I had about the future of 3D bioprinting is that, well, you talked before about how a transplant organ has a lifetime, and it won't last forever like our own organs. In comparison, I'm sure it's not anywhere close currently, but in the future, do you think 3D bioprinting, the organs will be able to last just as long as the original organ that's being replaced, or do you think it will have to be replaced on a basis such as like 10 years or 20 years?
Well, this is something we will have to learn. I would say we don't have the answers to that yet. I'm hoping that, of course, we can get a longer lifespan for the patients, but this is only a wish. And of course, I'm hoping that if you have the right materials and the healthy cells put in there, that we can do it. But this is something that only time will tell.
Yeah, and I'm sure even if you do have to get it replaced, just being able to have such a personalized medicine, it would be easier, and it can be printed on demand. But yeah, anyway, we discussed several different applications of 3D bioprinting today, and how the development of biomaterials and bioinks are key to making advancements in this type of medicine. It's clear that 3D bioprinting is a very revolutionary idea that could completely change how we do treatment in medicine. So what advice would you give for material scientists who want to pursue a career in this industry?
I have to admit that I'm personally very, very passionate about this area, because we can actually make a difference. Every day we are in the lab, we do make a difference, because every advancement we do in the biomaterials will actually help us learn to understand what it takes to take the next steps to bring it to clinic. It is an interesting and very exciting field. It's definitely an area that has a large potential to impact the future of health. And if you're specifically looking to work in the field of 3D bioprinting, learning about biomaterials and tissue engineering and 3D cell culture is definitely a good idea. Maybe studying biomedical engineering and performing internship at companies or labs who has bioprinting as their core application. That's the advice I can give.
That's great, actionable advice. So we really appreciate that, and we appreciate you coming onto the show today, Itedale. It was a pleasure having you, and I definitely learned a lot, and I feel super inspired, and I can't wait to see what CELLINK does next.
Yeah.
Well, thank you. The pleasure is definitely on my side, and I'm hopefully looking forward to meet you in real life, hopefully. if
Yeah.
that ever happens again.
I'll cross my fingers.
Yeah.
Thank you for tuning in to this episode of the It's a Material World podcast. If you enjoyed the show, consider subscribing and hitting the like button down below. And if you'd like to support us on this journey or simply show off your love for MSC, we actually released merchandise. And if you'd like to check out more of the designs, visit itsamaterialworldpodcast.com/shop or use the link in the description. We'll see you soon. And in the meantime, go change the world.